Wonderful. Thank you so much. I'm just so honored to be back here today. I really appreciate the opportunity to introduce the old colleague of mine, Christoph Simon. I hope everyone can now see my introductory slides. Christoph Simon has published over 120 peer-reviewed articles with more than 18,000 citations in major physics journals and other high-level international scientific publications, including Science, Nature, Physical Review Letters, Reviews of Modern Physics, the Journal of Modern Optics, Optics Express, the New Journal of Physics, and PRX Quantum. He received his education in Paris, Innsbruck, and Vienna, carrying out work with Anton Zellinger before moving on to fulfill postdoctoral research appointments, most recently, including his president appointment. Most recently, before his president appointment, he worked in Geneva, where he was the theoretical group leader under the supervision of Nicolas Jezin. Before that, he served as a research scientist at CNRS Grenoble, as well as a previous cross appointment with the University of Oxford and beautiful Santa Barbara, California. His most notable achievements in international journal publications, including entanglement purification for long-range quantum communication, near optimal quantum cloning of a single photon for an arbitrary input state, solid state light matter interface for single photon quantum memories, and he's surveyed prospects for intercontinental entanglement networks in the Review of Modern Physics. Other cutting-edge works include the entanglement swapping of independent photons, noiseless photon frequency conversion intended for hybrid quantum networks, observation of entanglement between microscopic and macroscopic photon ensembles, non-demolition measurement of a photon. I'll add, it's hard to measure a single photon without destroying it. And finally, the observation of entanglement between multiple macroscopic objects. Concurrent to these discoveries has been the development of technologies to roll out a global quantum internet for secure communication and having the potential for global quantum information processing applications, including satellite-based tests of theories of quantum gravity. But that's not all. He's written more physical review letters than you can shake a stick at on topics including violations of Bell's inequalities, quantum cloning, quantum repeaters, loophole tests of Bell inequalities, an atomic frequency comb for a memory of light, and spectral multiplexing for scalable quantum photonic systems. The list goes on and on. After an already auspicious career developing concepts and technologies for long-range quantum information processing and communication, he turned his attention to the area of quantum biology, developing a theory of optical communication in the brain, and a growing body of work introducing radical pair chemistry to a wide range of physiological phenomena, including the effect of lithium on hyperactivity, xenon-induced general anesthesia, the effect of weak magnetic fields on neurogenesis, and even microtubule reorganization. My supervisor, Philip, has encouraged me, and I'd be remiss if I didn't take a moment to introduce the radical pair mechanism to the uninitiated in the nuances of spin chemistry. So if you're asking, what is this radical pair model? I'll just take a moment to say, in a prototypical example, you would have something like a flavin, adenine dinucleotide, which I've labeled here FAD, and a set of tryptophan molecules, which can undergo an electron transfer from one of the tryptophans labeled W to the FAD. Now, once you've broken an electron away from a tryptophan and added an extra electron to the FAD, they can undergo what's called singlet-triplet interconversion. What's interesting is that the singlet state can easily recombine to reform the original state of the system, whereas the triplet state more naturally escapes. What can happen now is that if the electron escapes, then you can produce something like a free radical, a superoxide, that's an oxygen with an extra electron attached to it, or any number of radical scavenger species, as they may be called. Now, this becomes very interesting because free radicals are not only related to things like oxidative stress and aging, but they can also produce UV electrons during the photoemission process, which can be implicated in microtubule dynamics. Now, there's a whole wealth of phenomena that can go on in these processes. UV light has been implicated in microtubule reorganization, and the radical pair mechanism is related to this, but this is only, in some sense, a toy model because there's a wealth of other interactions that go on here. EED, I've labeled as electron-electron dipolar coupling, and of course, once you've produced a scavenger radical like radical oxygen species, it too can react with any of the initial parties, producing a wealth of rich chemistry with the potential for interactions on microtubules and other areas of physiology, as I mentioned on the previous slide. But I'm not here to talk to you about all that today. With further ado, I'd like to introduce Christoph Simon, who is going to tell us about his work in this area and prospects beyond. So please take it away, Christoph. Thank you very much.
Thank you very much, Nathan, for the detailed and kind introduction. So yeah, thank you very much for having me. It's an honor and a pleasure to tell you about these things. So yeah, I think Nathan showed you my title. So there's going to be radical pairs, there's going to be photons, and it's related to the brain. I thought, and here is a photo of Calgary, by the way, which I think is nice. I thought I would sort of structure the talk a little bit following my own history in this field. So as Nathan mentioned, I'm a quantum physicist, right? Or originally, I did a lot of work on foundational questions in quantum physics and then applications of quantum physics. And somehow about 7 years ago, I decided that I really wanted to try to understand the brain now and try to figure out if, to what extent these quantum phenomena are important for the brain. And that has led us now to some things that I think are important more generally in biology, or might prove to be important more generally. But this has sort of been the guiding question for me and for my group in this area. So it starts with this concept of entanglement, which is maybe the fundamental concept really in quantum physics, so certainly one of the really fundamental ones. So it's a unique quantum property that allows two or more particles to act in correlated ways, even if they are potentially, they can be very far apart, but they still behave in some sense as one coherent whole. There's a really holistic aspect to entanglement. We know really well, thanks to John Bell and many experiments since then, that these quantum correlations cannot be explained classically. They cannot be described by what we call local hidden variables. It's really a unique quantum thing. And the question that sort of drives me is whether that might be important for the brain. Why? Okay, so what would that accomplish? I see two main sort of reasons for taking this idea somewhat seriously. Like there's one is sort of an engineering reason, and the other one a philosophy reason. So I'll start with the engineering one. As Nathan alluded to, and as I'm sure there is now quite a large field of quantum information processing, quantum information technology, which is based on the discovery that quantum phenomena can help us process information better in various ways, right? There's quantum computers, there's secure communication. So there are advantages to using these quantum phenomena. And so it seems believable that if we have figured that out, maybe nature found that too, and is somehow using it in its own ways. So I think that's reason one, and it's, I think, in itself fairly compelling. But then there's another line of thought that has to do with consciousness, or subjective experience. Let me try to be clear. So there's a real mystery. How does subjective experience arise out of the brain? Out of all these neurons and molecules that we have in our heads. It is really still, completely unclear how that works. One of its most mysterious features is its unity in some sense, right? Like we have at any given moment, we have one stream of consciousness somehow, right? Like in some sense, one experience in our minds. Like how does this unity come about? And somehow the hypothesis or the intuition or the hope is that maybe that has something to do with entanglement, with this holistic character. So I'm not the only person who came up with this idea, but I find that very somehow intriguing and compelling that might be the case. So those are my two big reasons. Okay. Now, how could that possibly work, right? Like if I look at this, let's say we are trying to, find a quantum computer or something like that in the brain. Now, putting on my hat of like quantum engineer again, how would I do it? If I was nature, like what ingredients would I use? And there, I came up with these two, that are photons and spins. Those seem like to me like the obvious ingredients that you would try to use. Like we know that photons are the system of choice for sending quantum information over any kind of macroscopic distance. There's really no alternative, I think, to photons that we have discovered at least. So we should certainly keep those in mind. And then on the other hand, spins are really nice degrees of freedom. We know that spins can have, great quantum properties, even in condensed matter, even at room temperature or body temperature, like we just know that this can work from our quantum technology experience. So, okay, so those are my two sort of guiding themes then, photons and spins. And let me start, and I started historically with photons, so I'll do that here. So there are photons in the brain, and more generally, the cells, as I think most of you know well, cells emit photons, like we often call them biophotons. This is just one example where you can see biophotons being emitted by a brain slice, but people have done this also in vivo, with mice. And we recently did an experiment with collaborators in Ottawa where you can really just put the mouse into look at it with an EMCCD camera or something, you can see the photons, right? So this is a real thing. It's not super well understood where they come from at this point. It is thought that, and there's evidence, that the mitochondria are important, that reactive oxygen species are important. That's going to be a recurring theme in my talk, reactive oxygen species. But we really should, try to understand the sources and the detailed characteristics of these photons much better than we do right now, and we're trying to do that. The difficulty is that the rates are quite low, and that makes them hard to study, but not impossible. Now the question that I want to ask is whether these photons could be used for communication, in particular, in biology in general, but like in particular in the brain. We know that potential detectors exist. For example, opsins that are light-sensitive proteins. We have those not only in our retina, we have them also deep in the brain, and they seem to be doing relevant things. So there may well be detectors there. My own focus, as I said, is on quantum communication. Somehow I'm trying to distribute entanglement or something. But even classical communication by light would be very interesting, right? Like even if it turns out to not be quantum, just like classical photonic channels, that would already be amazing. So, okay. When I started thinking about this, about the potential role of photons, and in particular in the brain, I think it becomes clear pretty quickly that in a system as complex and sort of seemingly messy as the brain, you would really want a way for the communication to be directed, as of course, we know that the electrical signals in the brain are very specific and very targeted thanks to the axons, right? Because they are kind of like little cables that connect our neurons. So to me, it seemed pretty obvious then to ask whether these axons could also serve as cables for the photons, so as light guides. And I was excited to realize that that might really be possible. So the first thing you have to sort of look up is the refractive indices of the relevant materials, which in this case are the axons themselves, and then the myelin sheath that surrounds many axons. Many axons are myelinated, and you find that indeed the refractive indices of these structures are higher than those of the surrounding medium. So already the axon is higher than the watery medium that surrounds them, and then the myelin sheath is higher still. So at first sight, that's really promising. That seems like you have a waveguide right there. And then what we did in this paper in 2016 is like we pushed this a lot further. We did some really detailed modeling. Our biggest worry were probably the Ravian nodes, because there the myelin sheath is interrupted, and it's a bit of a complex structure with many layers, and there's scatters there and so on. So we tried to make a pretty realistic model of these Ravian nodes and convinced ourselves somehow that the light can indeed go through. So here we are looking at modes of light that are guided primarily in the sheath, and then we're looking at how these guided modes pass through these Ravian nodes, and the conclusion was that it seems okay. There is some loss, but like it certainly seems possible for the photons to go through these nodes and continue in the sheath. And then we did many other things. We looked at because of course axons are not actually optical fibers, right? They are not like perfectly homogeneous and smooth and engineered, right? They sometimes are a little bumpy, and their cross sections can vary and things like that. So we wanted to know how strong an impact that has. And again, like for all of this, so we did this, and here's the cross sections that are not perfectly circular, and in all of these cases, the conclusion was, if it's not too bad, like within biologically realistic ranges, this can all work like pretty well. So that was very encouraging for these general ideas. So the conclusion was that like pretty high transmission, a few percent to almost 100% seems possible in the relevant range, which is millimeters to centimeters, right? That's the big advantage compared to like real optical fibers, where we are trying to go hundreds of kilometers in the fiber. Here we're just trying to go a few millimeters or centimeters in the brain, and that seems really to be imaginable. A question that comes up is, I mentioned at the beginning that the rates for these photons are really quite low. That's why they are hard to study experimentally. It's maybe a photon per minute or something per neuron, or per second to per minute per neuron. But so is that enough? Could that be enough to do anything of interest? But I think yes, because that still means like billions or photons emitted per second throughout the brain. That could still connect. Again, there's sort of an engineering argument and a consciousness argument that one can make. Like on the one hand, if you have billions of photons, you can still wire up a lot of quantum systems. And we know from a quantum computing perspective that even 50 qubits is something that's really hard to simulate classically, right? This is where the technology is right now. It's at the level of 50 or 100 quantum systems. So you don't necessarily need billions of them even, to do something really meaningful that you couldn't do classically. And then the other thing is, if you think about consciousness, if that's your motivation, and sort of what's the bandwidth of consciousness, so people have tried to estimate that, and those are also quite small numbers, really. There's different ways of making estimates, but you come up with like hundreds to maybe a few thousands of bits per second of bandwidth for our consciousness. So it seems okay. Low rates, or relatively low rates, seem okay, really. Now, what about reality, right? So this is a nice idea, but like, is it borne out in any way by evidence? So I wanted, there's various things and various hints that this could be true, but I think here are two of the most relevant ones maybe. Like this is something on the left is an experiment we already knew when we, when we proposed this, where people had shown that there are cells in our retina that are called Müller cells that do in fact serve as light guides, as optical fibers. And these Müller cells are glia cells, just like the cells that make the myelin sheath. So that seems encouraging, so they are sort of similar in a material sense, they are similar cells, and they really do conduct light. So that was encouraging to us when we developed these ideas. Then on the right, though, is something better, so there's like evidence from after we made our proposal. This is an experiment by some neuroscientists in Canada, actually. And they see here, what you see here is a cross-section where you see the cross-sections of the axons, in fact, like these are these round little things. And one can see, so they are shining light on this slice, I think it's a slice. And you see that the myelin part, right? Sort of there's bright circles around the dark, around dark circles. So the darker part is the inside of the axons, and then the bright part, the light really seems to be going in the myelin sheath. So I think, and here is a zoomed in version of this, this is where the light would go according to our suggestion. And that seems to be kind of true, right? So it's not yet a super controlled experiment. I would still like to see experiments where one takes an axon, somehow prepares it carefully, one sends in light on one side, the light comes out the other side. So that hasn't really been done, but like because it's hard to do, but this kind of evidence exists now, where you can sort of see, I think, that the light really propagates in the myelin sheath. So that's, I think, not quite the end of my photon story quite yet. So here, as I mentioned, the existence of potential detectors, and I said that opsins seem like really promising, and that they are not the only candidate detectors in our bodies, but they certainly are up there. And this is a very nice paper from just last year, where they showed, so this is again in the brain, in the hypothalamus of rats or mice. It says it somewhere here, I can't find it right now. Mice, yeah. Where they showed that these neurons that are deep in the brain can be stimulated by violet light. They showed, and they do something biologically really meaningful in response, which is they regulate thermogenesis, right? So it is important for cells. There's always this question, do I do work of some kind, or do I produce heat? So thermogenesis is the heat production, and that heat production can be influenced by light. And they showed that it is sort of the receptor that is being used is this opsin 5, which is a light-sensitive protein that's related to the rhodopsin that we have in our retinas, but it's different. It's a different one. But so I think that's a sign. This is not, just to be clear, this is external stimulation. These are not biophotons that are doing this. This is like light that's coming in from the outside, but it is a sign that cells deep in the brain can respond to light in biologically meaningful ways. So I think that's also encouraging. Okay, so that was the photon part. So I said photons seem great for communication, but now I want to turn to my potential processors of information, which would be the spins. So it took us a little longer to find the line of attack for the spins, but then the first one that we found was anesthesia, which is nice, so anesthesia is obviously practically important, medically important, but it's also related to consciousness. So it was my obsession, as I explained, is somehow understanding consciousness. So it's anesthesia seemed like a really good starting point, and it's not really super well understood really to this day, you know, how anesthesia works. Of course, there's many things that are known about it, but sort of there's not really a coherent overarching theory of how it works. So my particular work here is going to be on xenon anesthesia. Xenon is an established anesthetic. It's not, you know, one of the most common ones, but it is approved for humans. It has some pros and some cons. But the reason that we were interested in it were some recent sort of physics type experiments on it. So the first one was this one here, and both of these experiments implicate spins. Okay, so the first one here is an electron spin resonance study on fruit flies undergoing anesthesia. So that was a really nice, impressive experiment that Luca Turin and co-workers did in 2014. They didn't do just xenon, they had a bunch of anesthetics, and they always observed a specific sort of difference in the ESR, so in the electron spin resonance signal, when the anesthetic was acting on the flies. And they had strains of flies that were receptive to the anesthetic and strains of flies that were sort of immune to it, and then there was a difference in the signals, you know. So it's a really nice experiment that showed, or at least strongly suggests, that some kind of electron transfer and electrons with, you know, free electrons with spins, something like that seems to be playing a role in anesthesia. So that was sort of point one. But then the paper that really got us going on this was this one here, where it's a Chinese group, an anesthesiology group, that showed an isotope effect for the potency of xenon as an anesthetic in mice in 2018. And they showed, so xenon has has a lot of isotopes, so they looked at 4 different ones. So they were able to show pretty clearly that this was really about the spin, so the nuclear spin of the xenon, and not, for example, the mass of the xenon nuclei, but really it was the spin that was important. So that is another sign, you know, because nuclear spin is quite, you know, it's a small little thing, you know, a small magnetic moment. It's really the only way, I think, that it can have such an effect is if it affects an electron spin that is nearby. And then the electrons, of course, are important for the chemistry, right? So to me, this is a clear smoking gun that, you know, electron spins are important, and then the nuclear spins sort of mess with them and have an impact through that channel. So that led us then to the radical pair mechanism that Nathan introduced a little bit before. So radical pair mechanism is a well-known, you know, concept in chemistry or spin chemistry. It refers to a pair of electron spins, often entangled to begin with, that are created either by an electron hopping from one molecule to another, or by a bond between two molecules breaking. Then you have these two unpaired electrons. The initial state is often a singlet state of the spins, but then the environment, so there's typically in molecules, you also have some nuclear spins around, you have an external magnetic field, at least that of the Earth, right? We'll talk about that more. So in this environment, then the spin states change, you know, because the spins are like little magnetic moments that sort of precess around these fields of the nuclei and then the external field, and that changes their state. That can change the initial entangled state, change its character, and then that changes what chemical reactions are possible, as Nathan was also briefly explaining, you know, because if the electrons are still in the initial singlet state, for example, it may be easier for them to recombine again, to form a bond again. But if their state has now changed because of this precession, then they may not be able to do that, and the products may diffuse, so other things will happen, right? And this radical pair mechanism so far in biology has been mostly discussed in the context of magnetic field sensing. There, however, it is very popular, so it's viewed as maybe the leading explanation for how animals like birds sense magnetic fields, but not so much has been done beyond. There hasn't been so much discussion of the radical pair mechanism beyond that context of magnetosensing. But we think that it seems to be much more general, and I'll give you a number of examples, in fact. But here, my first one is anesthesia. But in the meantime, we have applied this to many more things. So in the context of magnetosensing, again, people have focused very strongly on the cryptochrome proteins, which seems to be, certainly important in magnetic field sensing by birds. But even that point of view is evolving a little bit, it seems, just very recently, that maybe the cryptochrome is not sort of as core to it as people thought, but in any case, it's often mentioned, and it's certainly a famous example. It's also known to be implicated in the brain's response to low field TMS, transcranial magnetic stimulation. And this was in this paper here. When people talk about cryptochrome, they usually talk about two kinds of radical pairs. Like both of them involve a version of flavin, but so there's a flavin-tryptophan pair that's being discussed, and also a different flavin molecule superoxide pair. And I want to highlight here just that in the flavin-tryptophan pair, tryptophan is the donor, and in the flavin superoxide pair, O2 is the acceptor. Okay, I'll come back to that in a moment. So we like sort of knowing this context and all this excellent work on cryptochrome, then we had to think about how could it be, what could be the radical pair in the context of anesthesia, where we don't necessarily have those molecules, right? We don't necessarily have cryptochrome playing a role in anesthesia. Nobody has really proposed that. On the other hand, people have proposed or sort of concluded that the NMDA receptor is really important at least for xenon, and even a specific site. There's work on that. And it's known that site contains tryptophan. It's also known that reactive oxygen species are somehow important. They interact with the NMDA receptor. In Luca Turin's paper, people saw that they saw that oxygen had an important role to see these spin resonant effects. So that led us to suggest that maybe it's a different radical pair, in this context here, but with familiar ingredients, So we suggested tryptophan superoxide radical pair. Again, this is the donor. This would be the donor. The oxygen would be the acceptor, and that xenon somehow interacts with those. Our evidence for this specific radical pair is not strong. So it may well be that it's not that site, that it's not these constituents. We are really not sure at this stage about the anesthesia, but it's certainly it's a plausible possibility. I think that's what I would say. But the model is fairly general. Okay, but let me first tell you how they quantified the potency, the anesthetic potency. So they did that by what's called the loss of riding reflex. So basically you turn the mouse on its back and then you see if it can get up again. If it doesn't, then it's considered to be now anesthetized. And you check how much xenon you have to administer to reach that point. And you do that for different xenon isotopes that are listed here, some of which have nuclear spin, some of which don't. And these are the ones with and these without. And you can see that, the dose that's required is really clearly different. For the ones without nuclear spin, you only need this amount, and for the ones with nuclear spin, you need some other amount, right? So it really seems to pretty clearly correlate with the spin. And so then the challenge for us was, can we explain that with our radical pair model. So the radical pair model itself is fairly simple. You know, it's really, we focus here just on the dynamics of the relevant spins. So there's 2 electron spins in the model in this case, and then there is typically some nuclear spins. And in this case, we just took one dominant nuclear spin on the tryptophan, and then the xenon nuclear spin that somehow has to be nearby and interfering with the process, right? So very, very simple quantum model. We did want to, you know, get the physics right though somehow, right? So it's important, like in this Hamiltonian, you know, there's this coupling constants between the nuclear spins and the electron spins. So they are like really, really key. This is the external field, by the way. But then you have these coupling constants. So you know, we tried to calculate those and, you know, find believable numbers. So this is an oxygen, you know, this would be the superoxide molecule, the electron orbitals around that. And here's the xenon atom that, you know, approaches. And what one can see here, so this was done by density functional theory, which is sort of the quantum chemistry tool of choice for this kind of thing. And you can see that there is some electron population on the xenon. So there is some amount of transfer that happens of the electron from the superoxide to the xenon, and that is enough then to for the electron to interact with the nuclear spin of the xenon, because now it has some overlap with, you know, the nuclear spin of the xenon, and that's why this hyperfine coupling. It's a contact interaction. You know, that's how this interaction happens, and you get sort of a range of depending on exactly how you do the DFT, this functional theory. It's not a super precise science, but like you get an order of magnitude for how large this coupling should be. And then what we calculate is what's called the singlet yield. So I was describing before that it's all about the entangled state of the 2 electrons, right, in the radical pair, and that the nature of this entangled state changes from singlet to what's called triplet, so different kind of entanglement, and so the percentage, you know, this is this change that's being quantified by this singlet yield. You know, how much is there of singlet and how much of triplet, and that's going to depend, and that then influences the chemistry, and that depends on these rates, reaction rates between the spins and the spin relaxation rate, which is the rate at which sort of the quantum character of the spins gets lost in some sense. So those are important parameters. And then we just tried to see if we could correlate the so the idea, since it's the singlet yield that determines the chemistry, we thought, okay, this should be the quantity that correlates with anesthetic potency, because like the anesthetic action, that's some kind of chemical reaction that's important, you know, like maybe it's that chemical reaction that is influenced, you know, by this singlet triplet dynamics. So can we have a singlet yield that has a similar behavior to the potency? And the answer, so I'll spare you all the gory details here, but the answer is basically yes. You know, so we can find models or parameter values such that our model reproduces the difference in potency for the different isotopes of xenon. This is what this shows. There's sort of allowed regions in parameter space where we get the right behavior for the isotopes compared to the potency, or where we can reproduce the potency results. And then we also make predictions. So I mean, it's nice to reproduce something, right? But then to do real science, you want to do more than that. You want to say, okay, and here now my model also makes a new prediction that somebody could go and test. And then in our case, that's the magnetic field dependence. So like if you have a radical pair mechanism going on, you can certainly get isotope effects out of it, and that was sort of our first point. But then generically, you're going to have some magnetic field dependence, in this range of a few millitesla. And here's, for our model, that's what we predict. It should be different for the different isotopes, but like even if you just have one isotope, you should see that there's a dependence on the magnetic field, and that's the key thing that we would like people to test. We would like people now to do experiments on anesthesia with xenon to begin with, and see if the anesthetic potency depends on the magnetic field, and if it does, then that would be a very nice smoking gun that something like this radical pair mechanism is really happening. So yeah, so that's the conclusion on the anesthesia part. So we have a simple radical pair model that can reproduce what people see in terms of isotope effects. We have a specific proposal for what the radical pair might be. There is definitely challenges with that specific proposal. So for example, naively, one would expect the spin relaxation of superoxide to be higher than what we need for our model. But then that's something that the magnetosensing community had also encountered. I told you that they are also thinking about superoxide as one of the possible prayers there, and they have thought about this a lot, and they have come up with various ways to overcome this problem potentially. Yeah, anyway, so there's different ideas, like it might be immobilized, or other spins might come in and scavenge. Nathan is actually a big expert on the scavenging. He has done work on this with Daniel Kapnick. So similar things could apply here. But I'm not completely wedded to this particular radical pair model. I think the important message is that a radical pair mechanism seems to be a good candidate sort of explanation for what's going on there in anesthesia. And we have predictions. We predict magnetic field dependence. So that was our first foray into this with the radical pair mechanism. But since then we have done actually a lot more, because we found that this seems to be much more general somehow. So the next thing, so I'll go faster on those topics, but the next thing we did had to do with lithium, which is a very common treatment for bipolar disorder. So people study it quite a lot, also of course with animals, and people had seen an isotope effect already in the 80s, and there was a recent experiment now in 2020 where that was reproduced. So there's an isotope effect for lithium, for the effectiveness of lithium in treating hyperactivity. So to us, that makes it's a little bit less clear that there's only two isotopes, so maybe it could be a mass effect or something, but to us, since we were already doing, spin things, we said, okay, let's see if we can understand it. And the conclusion was, again, yes, like we can make a model that a radical pair model, even in this case, even Cryptochrome seems to be somehow implicated. So one can even go with the sort of standard mainstream radical pairs here. One can absolutely reproduce the isotope effect. And again, we predict a magnetic field effect. So this is something I have discussed with Doug Wallace, who's in the audience here. So he was already interested in trying to measure these isotope effects and sort of suggesting that they should also try measure magnetic field dependence and see, and again, if that is observed, that would be a sign that this radical pair dynamics might really be important there in this system too. So I'm excited to see if that works out. But there is more. So the work on lithium led us to the circadian clock, because there's a connection between bipolar disorder and the circadian clock. That's maybe not crazy, that like we somehow know that our daily rhythm and so on has a strong influence, right, on people's moods and so on. So in any case, the circadian clock is implicated, and there is, in fact, magnetic field effects that people have seen on the circadian clock. that is an area, a pretty active area of study. Now, what is nice about the circadian clock is that people have studied it a lot, so they understand the underlying sort of chemistry of the oscillator, of the chemical oscillator quite well, and there's quite well developed even mathematical models for that, which is something we didn't have for anesthesia, for example, right? But here it exists. So we said, okay, so that's great. So we can try and understand, like in more detail, how nature goes, all the way from the radical pair dynamics to then the macroscopic sort of circadian clock. And so we wanted to try to do that. And again, the short message is it works. We can make a model that has the quantum part, sorry, too far. Sort of the quantum part is this radical pair mechanism, and then we plug that into the biochemical model for the circadian oscillator that people have developed, and we can really show that we predict that the magnetic field, through its effect on the spins, then changes the period of the circadian oscillator in a way that's consistent with what people have seen experimentally. So we were excited about this. So that's a preprint. And then finally, oh no, I think I have two more actually. Then we also, so basically what we are doing now is we are going around looking for magnetic field effects in biology and trying to see if we can understand them with our, concept, which is the radical pair mechanism. And we have a lot of success with that, to our minds at least. So here's another one, where people have seen magnetic field effects on microtubers, in particular on microtuber organization. That maybe that microtubers, they like to organize themselves in a certain way. That's what they do in cells. They form these structures. And if you even just shield the geomagnetic field, that causes problems with microtuber assembly. So that's an experimental fact. And again, we contend that we can explain that with a radical pair model, because where you can see that there's actually a strong effect just when you turn the field off, right? So the geomagnetic field is somewhere here, and then it really drops down. As the field goes to zero, there's a fairly dramatic change that we think can explain. Again, in this this curve here, it says microtuber density, but it's really in our model that's related to the Singlet yield of this radical pair mechanism, right? The idea would be that somehow there's an important chemical step that involves a radical pair, and that is influenced by the magnetic field. And then finally, there's this paper here. So by the way, which I think, Nathan already mentioned that, he's interested in microtubers, and many people are interested. Microtubers are also implicated in anesthesia fairly convincingly. So we suggest that there may be, but people mostly focus right now on sort of electric phenomena with the microtubers, but we think that there may be important spin phenomena, in fact, that are happening there, and so that could be a bridge between maybe these two communities, in fact, and these sort of groups of ideas. And then finally, here's another really fascinating paper, experimental paper from a year or two ago, where they showed, so this is again just shielding the magnetic field, so not applying a strong field, just taking away the Earth's magnetic field that is there, has very significant effects on cognition in mice, and they showed that was mediated by reactive oxygen species, and they showed that and neurogenesis. So somehow the magnetic field changes neurogenesis, which then through reactive oxygen species, which then has a significant impact on cognition. So and again, we think that we can explain that with a radical pair model. Okay, so in this case, it's a Flavin superoxide model, And again, the key thing is this like characteristic dependence that you get when you go to low field, like when you really turn the field off, you have a pretty significant change in the triplet yield, and we can make that consistent with these observations. All right, so here comes my overall conclusion. So on the one hand, I talked about photons and the idea that photons might be guided by axons in the brain, might be somehow communication, an important communication channel. And I did mention, just to highlight that reactive oxygen species are thought of as likely sources for these photons. Then on the other hand, I said a lot about spins. So in particular, we started with anesthesia, right? That these radical pairs, the entanglement of these spins in radical pairs, maybe what is disturbed by the anesthetics. So maybe this entanglement is important, and then the anesthetic somehow messes it up, right? So that would be idea as far as anesthesia goes. But then we just saw that this seems to be much more general, right? These radical pair mechanisms seem to be important for many things, in fact, in cells, or at least it seems that way. So ranging from like lithium, for mania, the circadian clock, microtubules, neurogenesis, right? Like so many things. So it really seems like that it's an important sort of general thing that's happening in cells, where the magnetic field can influence, biological dynamics. And often, like I think in all these examples, we somehow reactive oxygen species were implicated, which are also implicated here, right? So that makes me really interested in these reactive oxygen species, because they could be a bridge that I am looking for. Now coming back to my original motivation, which was, let's look for quantum networks in the brain. I would really like to have a connection between the spins and the photons. And it seems that, reactive oxygen species like superoxide can have spin, but they can also, singlet oxygen, for example, can emit photons. So like maybe that's this interface, that could make it all come together. That's of course speculation, but it's at least, so the results are at least consistent with the idea that some kind of large scale entanglement that could maybe be created by these radical pair processes and then distributed by the photons could be important for consciousness. And with that, let me just thank my various people. So I have group members, former group members. I have really great collaborators. I wanted to mention maybe the most recent one. So on the radical pair, maybe two groups of collaborators. So on the photon side, we are collaborating with Daniel Oblak in Calgary, who is an experimental quantum physicist, but also with neuroscientists here at U Calgary. And then on the radical pair side, we just started a new collaboration with Wendy Bean's group at Western Michigan, who is doing very interesting things on magnetic field effects and reactive oxygen species in worms. So these guys are using frogs, just for the record, and here, this is Planaria, like flatworms. Little did I expect that I would be learning so much about these little creatures, when I studied quantum physics, but here we are. So thank you very much again, and I look forward to the discussion now.